First of all, I'm really happy to see the great outcome. Really happy to see that so many people are interested in 3D cell culture. It's a really important field. It actually has been around for way longer than I would have thought. It's been around for, I think I found a paper 20 years ago or something. I have been working in cell culture pretty much all my scientific life. So as Vanya mentioned, I was a postdoc at U of T here, did a lot of cell culture work there. My background is actually neurodegenerative diseases, kind of the opposite of oncology, because our cells are dying. Your cells cause death in the patient because they're not dying anymore, unfortunately. But I'm also very, very, very heavy background in imaging. And this is why I started with biotech as a technical and application scientist. So biotech, to just give you a quick overview in case that you don't know biotech yet. Biotech has been around for 50 years. In fact, we just celebrated our 50th anniversary last year. We're based out of Vermont, so everything that you would get from biotech is actually out of Vermont, except for me. I'm from Germany, as I said. Biotech is the market leader in plate reading and detection, liquid handling. We also do a little bit of robotics, and we have a great, powerful software, but we're also very, very heavily involved in imaging and microscopy. So that's what I want to focus on today. And again, I'm really glad that Dr. Souza gave such a great introduction into 3D cell culture work and how to actually create these structures. Can I just quickly ask who is currently working in 3D cell culture with 3D cell cultures? One person. People are planning to work with 3D cell cultures in the near future. At Biotech, we do have 2 automated microscopes. When I speak of automated microscopes, I'm talking about that you won't find binoculars with our instruments. You will actually, as you can see here, so these 2 microscopes I'm talking about belong to the Cytation family and the Lionheart FX. As Vanya mentioned, there's a Cytation 3 here at BioLab. I'm mentioning right now Cytation 5. That's our flagship at Biotech because it really nicely represents our whole portfolio, and I will get to that in a second. And when I speak about automated microscopy, so this is really important. Dr. Souza mentioned high throughput imaging. You don't want to sit down with a 1536 well plate and go through every single well by yourself, right? So you actually want to have something that does the work for you. Our Cytation family, and I'm going to talk more about the Cytation 5 in a second, and our Lionheart FX microscope, they do these jobs for you automatically. And in fact, they have been specialized on 3D cell culture. I should mention at this point, we do have our own wet lab in Vermont. So our scientists, PhD scientists, they stand in the lab just as you do. They use our instrument and they try to just run experiments as you do, but they try to optimize our systems for this. So this is how I can actually tell you that 3D cell culture has been optimized on our instruments. And again, I want to actually focus on the Cytation 5 for a certain reason today. So first of all, a lot of people, when I introduce this instrument to customers, to people who are interested in, and I show this picture and everyone looks at it and says, a microscope? Because I always say, we have a great microscope here. We'll do all the work for you, especially high throughput work. And everyone's like, Anna, that's not a microscope. This is a plate reader here. You can put in your plate here, and then you go and read it. So, technically, that is correct. The Cytation family, and in this case, the Cytation 5, it is a microplate reader, but it can come with a microscope. It doesn't have to. That's one of the important things about biotech. We do actually offer customized systems. But the Cytation 5 can be equipped with a microscopy part. And I just want to quickly show you the insights of this instrument because it's my personal favorite. If I had been working with this instrument during my postdoc, it would have saved me so much time just because it brings everything together. And I know this looks a bit much and a bit confusing, so let's break this up. What you can see here on the top, right here, is the plate reader part. So you would put in your plate anything from 6 well to 1536 well plates. You can also put in other labware. I will show you some of the labware that can be used with the system in a second. And then you can just do your normal plate reading. your fluorescence intensity, your absorbance, your colorimetric assays, etc. But you can also equip this instrument with an inverted microscope. And what you can see here is, so it is on the bottom here. So this instrument can be equipped, I'm trying to show this for both sides, I'm going to stand here, with a fluorescence cube holder that holds up to 4 fluorescence cubes. That doesn't restrict all the colors we have. We actually have more than 19 filter cubes that you can use with the system because some people just don't want to use DAPI, GFP, and Texas Red. Some people want to do something more advanced. So we do have over 19 filter cubes, but at once you can fit 4 in this filter cube holder. Up to 6 objectives. Very important. There are a lot of automated microscopes out there. Our microscope, keep that in mind, can hold up to 6 objectives, 1.25x up to 60x. Air objectives. So this is one of the differences between the Lionheart and the Citation. The Lionheart actually can fit up to, or can be equipped with up to 100x oil immersion. The Citation 5, for several reasons, only air objectives. And then you can get beautiful images such as this fluorescence image you can see here. But the Citation 5 obviously can do more. You don't want to only put in your cells and look at fluorescence, maybe you want to look at morphology. So the Citation 5 can do bright field, it can do phase contrast, even color bright field. We have a very high resolution color bright field camera in there. Or the camera does both, grayscale and color bright field, but it's very high resolution. And as I mentioned, fluorescence. And you can overlay these fluorescence channels and get like your beautiful images. And that's something that's very important to me to mention is when I come to this slide. A lot of people talk to me about, oh yeah, we just had someone else in and they asked us if we want to save some money on the objectives because there are objectives that you can get from the manufacturer of Olympus, for example, or Zeiss. But they're a bit cheaper because they're not labeled as an Olympus or a Zeiss objective. They're not the same. Even if they're from the same manufacturer, there's no way that these people would just say, hey, let's sell these objectives for half the price and it's the same quality. And because I'm imaging, like I'm a microscopist, imaging is so important to me. If there's something you want to save on, never, ever, ever save on the quality of your optical components in your microscope. If you go with biotech instruments or with others, just make sure you get proper optical, high quality optical components for your microscope. And biotech, so that's the only component that is not manufactured in Vermont. Because we think that for objectives, Zeiss and Olympus, they do a fantastic job, pretty much the best objectives you can get on the market. And for filters, for fluorescence, we use Semrock filters. And that's really important to me. Keep in mind, don't save money on these components. The lab where you can use with the Citation 5, as well as with the Lionheart effects. You can technically use anything. Except for the Citation 5, not a T75 class, but let's say you want to look at a T25, you can use this. You can use your cell culture dishes, and obviously any kind of microplate from 6 to 50 to 36 well plates. And that's when it actually becomes really important that you automate your system. Again, as I mentioned, I had a customer the other day at Sunnybrook who wanted to image 6 images per well in a 50-36 well plate. That is 10,000 images. I did the math, and I was like, oh my God. And he called me the next day and said, I ran out of space on my hard drive. I was like, yeah, no wonder, you got 10,000 images overnight. So keep in mind, this is really important if you want to go high throughput. And Dr. Souza also mentioned something else that was very interesting. So he was showing us paraffin slides from spheroids. But he also said something that's really important, what happens between 0 and, let's say, day 7. Really, really important. I worked as a postdoc with a lifestyle imaging system that was sitting in its own room. It was sitting in the dark all day. It was like a full room just dedicated to this one system. The Citation 5 is a fully capable live cell imaging system. You can, I mean, the system itself comes with a temperature control up to 65 degrees. I know that's a bit hot. Usually the 65, that's more for the plate reading parts, for plate reading assays. You wouldn't probably ever need to go that high for imaging. You can add gas control, CO2 and O2 if you want to. And then you can keep your cells in there happy and healthy for a pretty long time. If you want to add certain reagents, let's say drugs, and image at the same time, you can do that too, because you can actually connect two syringe pumps to the Cytation 5. And if it's fast kinetics, really important to keep in mind, inject and measure at the same time, or image at the same time. And something that the Cytation 5 can do and the Lionheart again cannot, that's why we have these two systems. It can be integrated with robotics, so it can be turned into an automated system. Either with our own robotics, so we do have a plate stacker, which is the BioStack. This holds your up to 50 plates if you want to. But for imaging, more important, I would say, is the BioSpa. That is our incubator, our automated incubator that sits here. Can be integrated with the system. It comes with a robotic arm. This robotic arm will take out the your cells on a microplate or on a dish. We'll actually put it into the Cytation 5 on a programmed basis. So you can just program it for up to, I don't know, people have done it for weeks. Automatically put it into your Cytation 5, and you can do fantastic kinetic stories. One of the important things with the Cytation, as I mentioned before, with these high quality components that you have in there, optical components, please don't expose those to humidity, because it will affect them dramatically over time. I know there's systems out there that have humidity involved. We did this on purpose. Cytation 5 doesn't come with a humidity chamber. You can easily image your cells over, I would say, 24 hours without having like evaporation of your media. There are also tapes that you can use. They're gas permeable, but they don't, the evaporation won't happen. If you plan on doing something very, very long term and you don't want to come in and every hour put your plate into your reader or imager, I would highly recommend using this Cytation. So the BioSpa holds up to 8 plates or other network and can be integrated with the Cytation 5, as I mentioned. And then you can do very long-term experiments.
That work for life cells?
Yeah. So this plate stacker, this is not in a controlled environment.
Okay.
So usually that's more for plate reading. So if you have, technically, if you could put it somewhere in there where there was a controlled environment, but then who hasn't incubated it that big? That's why we actually offer the BioSpa that holds up to 8 plates and pretty much is an incubator that will handle the plates for you. But I now want to talk about 3D cell culture, because I just wanted to give you an introduction into the system that we use and that is optimized on 3D cell culture. And again, at BioLab, they have the Cytation 3. It's the first member of this family, pretty much the same system. Only holds 2 objectives, but otherwise it's pretty much the same system. And now I want to talk about 3D cell culture, the challenges, why are we doing this, and what do you need to do beautiful images, but not only beautiful images, because you might also want to analyze the cells that you're imaging. I know Dr. Souza skipped over this thing. We all know about this. I had it in there because I kept it in here as well, because again, I did cell culture all my scientific life, and I never even came across 3D cell culture, which I'm now extremely embarrassed about. So I want to give you a quick overview why 3D cell culture is so important. Not only for oncology, but for other research fields as well, especially when you want to apply, let's say, drug testing. First of all, 3D cell culture resembles in vivo environment much better than 2D cell culture can do. Why is that? First of all, if you co-culture different cell types, they get together in a more in vivo physiological way, so they mimic the microenvironment better. There is a physiological cell-to-cell contact. Cells interact with the extracellular matrix, which doesn't really happen in the same way with a 2D layer. There are gradients for nutrients and oxygens. So for those of you who work in oncology, you know that there are, for example, parts of hypoxia in tumors. So it's really important to mimic this. There's also non-uniform exposure to test molecules, and that's when I'm talking about drug screens. And there's several zones that you can usually find in these 3D structures. And one of the things that I find even more astonishing when I read a few papers about 3D cell culture is, if you compare 2D cell culture with a 3D cell culture model from the same cell types, and I think Dr. Souza also mentioned this briefly, you have different gene expression. And that is really, really important. You can't just go in there and say this 2D cell culture layer that I have here is exactly what I'm looking at when I look into, let's say, a tumor in my patient. So it's not always necessary. It really depends on what you're looking at. But especially when you work in oncology and you want to, for example, test new drugs, really important, I would say, to start developing your 3D cell culture systems. And just as a reminder about the microenvironments that you can find in tumors, I actually have two here. You can see how different the different zones here are. So you have exposure to the extracellular matrix here. You have this non-uniform exposure to the drug molecule. Oxygen doesn't get to every part of this microsphere. So really, really important to mimic this, especially, again, for drug development, because yeah, in 2011, there were approximately 900 anti-cancer drugs that went from the lab into clinical trials. And only 12 of them were approved by the FDA because they showed in the end that they were not as effective in the patients as they were shown in the lab. And that's huge waste of money. It's a huge waste of time. Let's actually develop a better way of doing research in the lab. All right, what do you need to actually do really good 3D imaging? First of all, as I mentioned, well, if you have your manual microscope and you want to sit there and do your 1536 wells manually, you would need an autofocus. I wouldn't recommend that one. Our instruments obviously come with an autofocus because they don't have binoculars. You really need a proper one. Most systems use an image-based autofocus. So what happens there is the instrument itself goes through several focal planes and then picks the one with the highest contrast, because the image can actually determine the contrast in the the system can determine the contrast in the image. Disadvantage is a bit slower, and if you have very sensitive fluorophores, because it literally takes your samples and goes through it as if you were doing a Z-stack. So if you have dim fluorophores, you might actually cause phototoxicity, photobleaching. I will show you in a second a solution biotech came up with. So we do have this kind of autofocus, but we have even a better solution for it. I will give you an example of our autofocus in a second. Another thing really, really important, because I know a few systems out there do not have this ability, is not only Z-stacking, so taking several images, but also collapsing them in the end to get a Z projection. If you have a Z projection from a wide field microscope, so I should mention that our instruments are wide field microscopes, you will end up with a picture that looks a little blurry, because obviously from the different planes, you get also scattered light from other planes and look a little bit blurry. So another important thing that you should have is a good deconvolution algorithm in your software. And a lot of people I talk to, I have so many people coming to me saying, I want to have a focal microscope. And I always ask them why. And they say, well, because I want to create really beautiful images. And if that's the only reason you're looking into confocal microscopy, please don't do it. Deconvolution algorithms nowadays are so good. I mean, look at this picture. This was a collapse picture, and then we ran our deconvolution algorithm with our software. I did a comparison when I was still a postdoc, because at some point I didn't want to go to, I think I went to the AOMF, which is now, I think, the Mars building. I did a direct comparison between confocal microscope images and then just those that I took and had deconvolved. And I couldn't tell the difference. I would always recommend, if it's just for beautiful images, don't waste your time, because it takes so long to go and take confocal images. It takes a long time. It actually might cause more bleaching because your exposure time will be longer. Depending on the pinhole, the size of the pinhole, the more contrast you have, the fewer light gets through, the more signal you need from the sample. A wide field microscope won't care about that, and you get, like with this algorithm, really, really beautiful pictures. And then that brings me to the last step, analysis. Really, really important, because you don't want to only have these beautiful images. You want to be able to quantify. And I will show you in a bit how our software, Gen 5, actually does a really good job with that. And the reason for that is that, as I mentioned before, we're market leaders for plate detection, so for plate reading. That usually comes with a heavy analysis part of the software, where you can do like all the calculations with all the numbers. And we have the same software for our plate readers as well as for our images. So the analysis part is the same. So we have a powerful, powerful analysis software. Okay, I want to just like quickly check mark the requirements that I mentioned and that biotech actually provides. As I mentioned, we do have this autofocus that just like goes through your sample in different focal planes, looks for the highest contrast. We also have something which is called a laser autofocus. And that's something I'm really, really excited about because it is extremely fast. I will show you in a second why. It will leave your sample alone, so you won't have any phototoxicity, you won't have any photo bleaching. It is very accurate. If you work with something like 3D cell cultures and spheroids, and you go through the Z planes and look for the highest contrast, you might run into trouble if you use this kind of focus, because of the 3D nature of these samples. Not with the autofocus. The reason for that is the way the autofocus works. It has a very long wave laser that it shoots against the bottom of the vessel, whatever it is, Microplate, T25+, whatever. It determines the distance from the bottom of your vessel to the objective. It knows exactly where the bottom is, and then will take it just a step above until it reaches the focal plane and you actually tell the laser autofocus, this is where I want you to be. And it's extremely accurate. So you can actually determine your focal plane by just going through the same Z plane, saying, okay, this is my actual focal plane. Then you tell the laser this is the bottom of the plate, and this is the step you have to get higher than this to actually end up in this focal plane. So it only takes one step. It's just the laser, brings it right up. No photo bleaching, nothing. Very fast, very accurate, will always land in the same spot. Z-stacking and projection. So these are different images that were taken by the Citation 5 of spheroids. And you can see here, and that's what I was talking about when I was saying, if you have a wide field microscope, you will end up with scattered light from different focal planes. So you have this kind of like blurry image when you collapse them together. And unfortunately, they don't have just the collapsed image. I wish I could have shown you that. They actually took the image right away and deconvolved it. But you can see that the image actually looks really nice. So you get rid of this blurriness because of the algorithm that we use for deconvolution. And another thing that you can do, as I mentioned, our system is light imaging capable. You want to be able to do kinetic studies. You want to know how your cells are forming these spheroids. So we can provide these time-lapse videos or time-lapse images that can be in the end combined to a video, and you have a really nice way of following your spheroid forming. Your cells forming a spheroid. And then coming to the analysis, I think that is really important because I don't talk to many people who do 3D cell culture that say that they actually analyze these cultures. Because they say we just take the images, we maybe follow them, maybe kind of like figure out if they grow in size, if they shrink. With the analysis part of Gen 5, you can do a little bit more. So for example, this is the image that I showed you before, this one. So if you just take the DAPI channel, for example, you can do a cell count from this 3D Z projection. So these are all the cells that you saw from all the different focal planes that, someone else took an image from. So you can, for example, do a cell count. You can actually determine, so as you can see here, there's the size and area and fluorescence intensity. So you can either focus on the DAPI channel or any other channel, and you can actually do a really, really good analysis. Again, you can measure size, fluorescence intensity, etc. In this case, the focus mainly on the cell count, which is, in this case, 328 cells. So that's something that our software can do. So we can translate these gorgeous images into numbers, really, really important. Oh, and something I forgot to mention is you don't only want to image your cells. At some point, you have to take care of the cells too, of the spheroids. You have to maintain them in a proper way. And if you're not so lucky by having the magnetic system where you can actually fix them in the corner of the well and then exchange the media, let's say your cells are kind of like floating in the wells of your microplate, you might run into the issue when you change the media that you actually lose some of your spheroids. And this has been done by our scientists in Vermont. I don't know if they were just a little bit sloppy, but when they did the manual media exchange, they lost a few of their spheroids, unfortunately, because if you put in a pipette and you aspirate the media, it looks like it's a straightforward step, but actually it's really hard to control how fast you aspirate the media with a pipette. As I mentioned before, biotech is also a market leader when it comes to liquid handling. So that means plate washing as well as dispensing. This is the multi-flow FX. That's one of our strip washers that we have at biotech. And this strip washer can be equipped with something which is called the Automated Media Exchange Module, or in short, AMX. The great thing about this module is that instead of using any kind of vacuum, it uses a peristaltic pump to remove the media in an extreme gentle, extreme slow way. And when our scientists in Vermont did that, and then they tested their, I think it was a 384 well plate, they ended up with spheroids in every single well. So it was like very effective. I have a short video that I want to show you how the system works.
Biotech's versatile multi-flow FX multi-mode dispenser automates many routine liquid handling tasks. 3D cell structures, such as spheroids and tumoroids, require regular media exchange to maintain culture viability and encourage growth. Biotech's AMX module for multi-flow FX gently and automatically removes and replenishes media without dislodging 3D cell structures. Across a broad range of applications, the compact multi-flow FX has the versatility and performance to meet all your liquid handling requirements.
And as I mentioned before, you can automate the system, right? You remember that? So you have the BioSpa sitting here in a biosafety cabinet in case that you want to work sterile still. It can fit into a biosafety cabinet. Maybe not all of the standard biosafety cabinets because it's a bit deeper, but there are biosafety cabinets that can hold the BioSpa together with the Cytation and with the multi-flow. So you can have a fully automated system that will handle all of your cells automatically, again, up to 8 experiments at a time. It will let you know by text or email if your experiment is done. Oh, we have a question. Yeah, I'm sorry.
So does this only work with 96 or 384?
No, this works from 6 well up to 1546.
And is it, how about if you're changing media and the different wells have different conditions?
So
can you actually control that?
So actually, I'm sorry if we talk about the imaging and the BioSpa that holds any microplate because it just has to be in the right dimension. The AMX module, that like specific washing for just cell culture media exchange, that actually only does 96 and 384. We have other wash models that can do cell culture media exchange, but they're not specialized on the.
And can you like do for different wells, different maybe like for example, I saw that it was changing. It was not like sterilizing between each. So that means if you have different treatments that might get cross-contaminated. Is that an issue that can be addressed?
That's a good question. So we do have different options of dispensing. I think you're referring to dispensing. I mean, like if you do the media exchange, that's just media. So the multi-flow also can actually dispense certain reagents. That's not there only for media exchange. It can actually dispense, let's say, reagents like drug testing. You can use 2 peristaltic pumps and 2 syringe pumps on the system, which all feed in 2 different manifolds. So you can hook up to 4 reagents, and they won't cross-contaminate because the system won't go all the way for the aspiration, yes, but not for the dispensing. It won't actually touch it doesn't touch the medium, and you can dispense 4 different reagents without cross-contamination. So I just wanted to quickly show you how this is dangerous, how this system works together if you have in a biosafety cabinet. And as I mentioned, so the BioSpa has a robotic arm. It can take out in this case, it's a 96 well plate from one of these 8 drawers. It will delete it, really important. If you want to do the media exchange, so then it will bring it over to the multi-flow FX, which in this case, so these are is the media that is going to be used for the media exchange. Then the robotic arm will bring the plate back into, either will bring it back into the incubator, or it will actually take it from the incubator right into the imager. So you can, let's say, image for 5 days straight every hour, and you don't have to be around at night, or have your graduate students be there at night and do the work for you. Yeah, and as you can see here, and this whole system, again, integrates seamlessly. Can be programmed very, very easily. We have a field application scientist that will help you if you're ever interested in that. And then you get your beautiful images automatically. You can even automate the analysis, which is really great. So you can actually walk away, come back the next day. You have the images, they're processed, cells are counted as an example, and you will have your cells, your images and your analysis ready. And now I want to actually show you some of the spheroids that we imaged either in Vermont or by our field application scientists at demos at a customer site, or sometimes customers send in their images as well if they're really happy about the system. They provide us with our images or their images. So one of the images I want to show you is this HepG2 Spheroid stained with the nuclear ID in blue. So this was actually done in Germany at the Greiner Bio 1 facility. So they provided this image to us. Then we have a human HT29 tumor spheroid that was sent in by a customer. This was actually one of our contests that we have that is actually currently also running, where you can send in your beautiful images and you get a cash prize, and then you can also end up in one of our calendars. So this one was actually one of our calendars because it's so beautiful. And we have a neurosphere in 20x magnification with GFP expressing neurites. And then, as I mentioned, you can also use this for analysis. Never ever forget that images can actually be turned into numbers. Really important. So this is 3D liver microtissue hepatocytes treated with diclofenac. Diclofenac is an NSAID. Non-steroid anti-inflammatory drugs are known to cause liver damage. So you can apply these on in this case, your hepatocytes, and you can, over time, and with different concentrations, see that in green you see dead cells because they're stained with cell.screen. So you can actually follow the death of these cells, the damage of these cells, and you can count them in blue as I showed you before, and then you can actually analyze how many cells are dead versus live. And then we have a lot of more. One thing that I haven't mentioned was that you can overlay not only fluorescence images, but also bright field and fluorescence. That gives you a little bit more overview over the morphology of the cells. And I think you showed that picture. Yeah. This one. So yeah, we work with Greiner, as you know by now. We work with Greiner together. So yeah, I found this in our database, and I realized it's one that's also on the Greiner website. So we can do a lot of more. So again, these are images from us, from customers who send us these images, and that is pretty much what I have to say. Just as a reminder, because I said we have 2 systems. So the Citation 5, main difference between Citation 5 and Lionheart-FX. Plate reader, just the microscope. Temperature control up to 65, which is mainly for the plate reading assays. Temperature up to 40. This one has air objectives up to 60. The Lionheart can have also oil immersion up to 100. So a lot of people prefer to have the combination system because it gives you a lot of advantages. However, if you need, for example, up to 100x, then the might not be the right system for you. Otherwise, the software is the same, everything is the same, except for these main differences.